All right, are there any questions? Okay. So today we are going to get started with the plant biology portion, right, of this course. So now we have already covered basic molecular cell biology. Then I introduced you to a lot of engineering methods, right? You have already taken 2 exams on the biology portion, right? So now we are ready to get started with the plant biology, right? Now the good news is that, there are a lot of similarities, right? Like whatever we have learned about, organization of DNA, RNA, protein, transcript, those things are all the same, right? But there are some important differences, right? And so, things will be easier, but at the same time, I have to cover a lot of territory, right? And, the book that I'm using, I think we mentioned that in the syllabus, that's actually 760 pages long, right? I have read that book twice, and now I'm making the notes out of that, okay? So I'll try to cover the entire book, but only the important things, right? Picking up. So it has got like 24 chapters. I'm going to try to cover 4 chapters every day, right? Only the important things, okay? Because a lot of it is detail that we as engineers, we don't need to worry about, right? So hopefully I can get through that. And for every, so I'm basically going to be about only 1 lecture ahead of you guys, okay? So today I have to go back and start preparing for the next lecture. So bear with me, but I think we can make it. And I have invested a lot of time, but I think the information that I'm going to give you is going to save you time if you are interested in working on agricultural applications, right? So first let's start off with chapter 1 in that book. And the name of the book is mentioned in the syllabus. You don't have to buy the book, but later on if you have to refer to it, you can go back and refer. So chapter 1 is basically an overview chapter, and it's called Plant and Cell Architecture. Now, a lot of the things that are discussed in this chapter you already know, because whatever you learned in the context of molecular biology, right, in the first third of this course, right, basically that carries over to plants. However, there are some important differences, right? The first important difference is that plant life cycles alternate between diploid and haploid generations. What is a diploid generation? Anybody remember? What is a diploid? Diploid cell means 2 copies of each chromosome, all right? So if you're looking at animals, right? So if you're looking at animals, the way that reproduction occurs, all right, you're going to have 2 copies of each chromosome, so 2n, right? From that you will get the haploid gametes that have only 1 copy of each chromosome through a procedure that is called meiosis. Yeah, so you all guys already know all of this stuff, all right? All right? And then to get back the diploid organism, right, you're going to have 2 gametes of opposite type. There'll be fertilization, right? And then you will get back to a 2n organism, and then there'll be repeated mitosis. Okay, so that's the life cycle of an animal, right? For a plant, it's a little bit different, right? So for plants, first of all, plants don't have to be 2n. They might be tetraploid, okay, octaploid, and so on, right? They're not diploid all the time, right? But even leaving that aside, okay, let's say you have a diploid plant, right? Now, when this diploid precursor cell undergoes meiosis, all right, it will produce a haploid cell, okay? However, the haploid cell, it's not going to directly go to fertilization. It can, undergo mitosis, right, and produce a multicellular organism, haploid organism, right? So one end, then this undergoes mitosis, right? Then this will produce a multicellular haploid, right? And then later on, that haploid is going to produce, the multicellular haploid is going to produce the egg and the sperm, again, not by meiosis, because meiosis has already happened before, right? So meiosis happened here already, all right? But, so it's going to produce a multicellular haploid, and then that will produce an egg or a sperm, not by meiosis, but by mitosis, right? Because you don't need to halve the number of chromosomes. So by mitosis, it is going to produce the gametes, right? So that's why you have 2 different kinds of generations, right? In one case it is diploid, right? The other case you can have a haploid generation where things are undergoing mitosis, right? So that's one big difference between plants and animals. Okay, so keep that in mind. Any questions on this? And they have different names, like they refer to one of them as sporophyte generation, the other one as gametophyte generation, and so on. But again, I mean, as far as we engineers are concerned, all that terminology is not that important. If you want to read that, you can read in the book, right? So yeah, I can just mention the terms like gametophyte and sporophyte generations. Now the second important thing, right, important difference between plants and animals, right, is that in the case of plants, right, the cell is enclosed inside a rigid cell wall, right. In the case of animals, it will be just the plasma membrane, right. There is no cell wall, right. So the second important difference is that plant plasma membrane is enclosed inside a rigid cell wall. So typically if you have a plant cell, right, where you have the plasma membrane or the delimiting membrane, and what is this membrane made out of? Or what is it called? The cell, what? Lipid bilayer, okay? So this is the lipid bilayer plasma membrane, right? Right? But in addition to this, outside this, you're going to have a rigid cell wall, right? So there will be a cell wall outside. Okay, and that's quite hard, right? Now, there are a number of consequences of this. One consequence is that, if in an animal cell, all right, let's say you have a parent cell that is giving birth to other cells, okay, then, the descendants, they can freely move around, right? So you might have cells that originated from different parents, right, ending up in the same location, right, or in the neighboring location. In the case of plant cells, that's not going to happen because the cell cannot cross the cell wall of another cell and go in there, okay. So a consequence of this is that the stem cell cannot move around, right? Or the neighboring cells come from same ancestor. All right. Another consequence of this is that because plants have got rigid cell walls, all right, that means a lot of pressure can be built up, right? Let's say if the cell absorbs a lot of water, okay, the pressure builds up. So the wall is there to basically withstand that pressure, right? The cell wall is there. If an animal cell takes up too much of water, guess what will happen? It will burst, right? For the plant cell, that's not going to happen. And for plants, it is really important because the plant cell has to absorb water, okay? And the water from the soil has to go all the way up to the top of the plant, right? So there's a lot of pressure and a lot of dynamics involved, right? And the reason that I'm going to cover that part in detail too about how water flows in plants and all that. The reason I chose to do that is because you're an engineering audience, all right? So you know the basic laws of physics, all right? So how that transportation takes place, that should be quite straightforward to explain to you. Not again very relevant from the point of view of genomics, all right? But it's almost for free you can get that information, right? So one consequence is this, and then the second thing is that plant cells can tolerate large hydrostatic pressures. One other important difference between animal and plant cells is that the neighboring cells, okay, so you have the plasma membrane, right? Outside that you have the cell wall, right? Now usually if you have a cell with a plasma membrane, right, the plasma membrane is a lipid bilayer, right? So the inside and the outside of the cell cannot communicate directly, right? You have to cross the layer somehow, maybe through vesicular transport or some ion channels or something like that, right? But in the case of plant cells, yeah, you're looking at something. What did you miss? I mean, what do you, higher up or what? Where? Tolerate large hydrostatic pressures. Hydrostatic. All right, hydrostatic pressures. Yeah, yeah. You don't have to take the notes, but that's up to you. You know, because I said I'm going to go, but if you have trouble, understanding the handwriting, then that might be a problem too. But again, I mean, the voice recording is also there, so they can tolerate large hydrostatic pressures. Now, for plant cells also, you know, in the plasma membrane, there are openings, right? There are openings that are called plasmodesmata, right? That will allow the cytoplasm in one cell to communicate with the cytoplasm in the other. So neighboring cells, they can communicate that way, right? That's in the plasma membrane. Yeah. So it's all connected together, right? And yeah, let me just finish mentioning that. Then I think things will be clearer. So neighboring cells linked by plasmodesmata, right? So if you have two cells, right, that have plasma membranes around them, then they are also linked together, right? So let's say this is the plasma membrane outside, right? But they are also linked together. And of course, this is not drawn to size, right? They are linked together by these channels that are called plasma desmata, right? Which means, right, that the cells, so if you want to push something from one cell to the other, right, you have a number of different routes, right? You can either cross the plasma membrane, right? That would be like the transmembrane transport, right? Across the membrane, there are mechanisms for doing that. You could make use of the plasmodesmata, right? That is called the symplastic transport, right? And I will write these things down. So there will be symplastic transport. So this network of all these cells, the cytoplasm or the cytosol of the cytoplasm of the cells connected together through plasmodesmata, that is called the symplast, right? So molecules can flow, through these plasmodesmata, and that is called symplastic transport, right? On the other hand, you could also have apoplastic transport, okay? Apoplastic transport is basically even the cell walls, right, of the different cells, they are connected together, right? There are cracks and crevices in there, right? So without getting into, without crossing the plasma membrane or getting into the cytoplasm of a cell, things could be transported around, right? That would be apoplastic transport, right? Basically the transportation pathway that is formed by the cell walls, right? And then the third one is going to be the transmembrane transport. Yes. I mean, the cell wall will not be there, okay? The way it is laid out, probably a portion of the cell wall is digested, right? And then the connection is there, yeah. And again, I mean, those are details, and in the book they spell all those things out. I'm kind of skipping that because I have to cover a lot of territory quite quickly. So transmembrane transport, right? All right, now let's talk. All right, any other questions so far? Let's talk a little bit about the cell wall of plants. Okay, usually you will have a primary cell wall and also a secondary cell wall. Right. So cell wall properties. So you have a primary cell wall which is thinner, right? Which is thinner and more stretchable, all right? That cell wall is laid out as the cell grows, okay? So it has to be flexible because the cell has to be able to grow, right? So this is laid out before cell growth is complete. So this boundary has got to have a certain amount of flexibility, right? Now once the growth is complete of the cell, right, it has grown to the right size, then inside the primary cell wall, right, there'll be a secondary cell wall, right, which is much more rigid, right, and it's also lignified, right. So the secondary cell wall lot thicker and it is lignified. Lignified means there is some molecule over there, that is going to call lignin, that is going to contribute to make it really hard, right? So thicker and lignified, and this is laid out once cell growth stops. And obviously, the secondary cell wall has to be inside the primary cell wall, okay? Because cell is going to lay that out, right? I mean, so secondary cell wall lies inside the primary cell wall. Now, not every cell will be, remember, although this secondary cell wall is laid out, right? Maybe in a certain class of cells that secondary cell wall is not needed. So there might be enzymes that will chew up the secondary cell wall and you might see the secondary cell wall is not there, right? So there are differences, but this is just the overall picture, right? The most general picture. So as I mentioned before, rigid cell walls allow plant cells to build up water pressure, which is called the turgor pressure. All right, any questions so far? Now, see, in any multicellular organism, right, you will be having both differentiated cells as well as stem cells, okay? Because stem cells are needed because if you're going to produce new differentiated cells, all right, the differentiated cells themselves cannot divide, okay? So you need the stem cells. So in any tissue, you're going to have stem cells and you're also going to have differentiated cells. So some of those stem cells will again differentiate into the differentiated cells, some others will remain for future use, right? So in the case of plants also, most of the division, right, is carried out by groups of stem cells, right? And they are called meristems, right? So meristems, these are basically groups of stem cells. So these are cells which have the capability to divide. And they have different names, right, in different regions of the plant, right? So first of all, if you have a plant, all right, you're going to have a root, that is the underground portion, right? Then you have shoot above the ground, and then you have leaves, right? So these stem cells, all right, or these meristems, they have different names. If it's in the root, it's called the root apical meristem because it's at the apex of the root, right? And in this book it's abbreviated as RAM, right? So they are the ones, they are the stem cells from which the other tissues of the root and all that will grow, right? Roughly speaking, right? Then you also have shoot apical meristems, right? SAM. And then inside the shoot of the plant, right, you also have something that, a bunch of stem cells that are called the vascular cambium, right? See, in the plant has to be supplied with water and nutrients from the soil, right? So you need a network of pipes that will do that. And I will talk about that later, right? And at the same time, the leaves in the plant are the organs that do all the photosynthesis. They produce all the glucose, sucrose, and so on, right? And the roots also need nutrition. So you need pipes to convey that too, right? And the vascular cambium basically is a bunch of stem cells that give rise to those pipes that are needed, right? To the vascular tissue. So vascular cambium gives rise to vascular tissue. And we'll talk more about this in a later chapter. Many of these things that I'm mentioning, this is the introductory chapter, so I'm pointing out the differences and just giving you the highlights. Many of these things I will talk about later, right? Maybe not in a whole lot of detail, but at least some more detail than what I'm doing here. Right? And then there is something else that is called the cork cambium. That is also another group of stem cells, right? That will build up some of the stuff inside the shoot of the plant, right? Now, as you already know, most cell membranes, or actually all cell membranes are, no, I should change that to most. So, cell membranes are made of lipid bilayers, right? You already know that. Now, most of the membranes in the cell, right? So the lipid bilayer means there's going to be a lipid with a hydrophilic head, right, and two hydrophobic tails, right? You remember how that comes about, right? In the case of most of the cell membranes, it's usually what kind of lipids? You may not know that. Phospholipids. Most of them it is phospholipids, where there's a phosphate on the head, right? So most membranes are made of phospholipids, right? So most membranes are made of phospholipids. However, the membranes for plastids, which is the group of organelles to which chloroplasts belong, are made of glycolipids. That means instead of a phosphate, you'll have some kind of sugar over there, right? It's a head made of sugar. Now let's talk about some of the other, compartmentalized organelles that you'll find inside a typical plant cell, right? One group of organelles is what is called the mitochondria, right? So mitochondria is the site of what? Energy generation, oxidative energy generation, okay? So this is the site of, and that's true even for even for animal cells, okay? So site of, let's put aerobic, right? Energy generation. So that's like the powerhouse of the cell. Most of the energy is generated there. If you're looking at a plant cell, you're going to have chloroplast, and that's the organelle where photosynthesis takes place. And we will discuss photosynthesis in detail, right? In the next lecture we will do that. So site of photosynthesis, right? Now I'd like to point out that mitochondria and chloroplast, okay, they have two membranes, all right? It's not just one single lipid bilayer, there are two lipid bilayers, right, for each of these guys, right? So mitochondria and chloroplast each have two membranes. So keep that in mind. And this is important because uh, in the case of mitochondria, they have to generate energy, right? And the different membranes play different roles, right? But they each have two membranes, right? Uh, Now, in addition to that, there are some other organelles. One of them is called a peroxisome, right? And again, what is the function of a peroxisome? Huh? Yeah, reactions that contain hydrogen peroxide. So compartmentalize reactions that produce H2O2, right? Then in addition to that, if you're looking at a plant cell, you will have another organelle that is called a vacuole. These organelles, vacuoles, they store starch, right? So starch is basically a polymer of sugars, made up of sugars. All right? So these organelles, they store starch, and they will also compartmentalize reactants, right? For example, metabolites in response to herbivory. See, what is going to happen is plants have got natural defense mechanisms, all right? So if animals come and eat up plants, right, what is going to happen is that you have vacuoles, maybe some toxic chemical is stored inside the vacuole, all right? You go and disturb the plant, that stuff is released and it will mix up with something, make it really unpalatable for the animal, right? So plants have defense mechanisms like that. So vacuole can play a role in that part. Now some terminology. The vacuole again is a compartment that has got a membrane, right? And the membrane of the vacuole is called a tonoplast, right? So membrane of vacuole is called a tonoplast. Now another type of organelles that you will find in plants, they are called glyoxysomes. Glyoxysomes. And these actually carry out the metabolism of a compound that is called glyoxylate. And this is something that we will encounter, right, when we discuss photosynthesis, right? Because photosynthesis has got quite a few details in it, okay? And I'm going to try to do that here because there are places where you can tinker around to increase biomass and so on, okay, of the plant, right? So we need to understand photosynthesis. So this is something that occurs. Glyoxylate is something that occurs in photosynthesis in the carbon fixation reactions. Right? I will talk about that later, so don't worry about the terminology. But glyoxysomes are compartments where those are metabolized. Right? Then there's only one more compartment that I want to talk about, and these are called oil bodies. Right? These basically store fat, right? Or they'll store triglycerides. Again, what is the composition of triglycerides? You have a glycerol backbone and three fatty acids, right? So if you have an oil body, right, so that is going to have a membrane around it, right? So what kind of membrane is that going to be? Remember, for the oil body, right, it's triglycerides, that means three fatty acids, right? So it does not have any hydrophilic region, right? So if it's going to store triglycerides, what kind of membrane is it going to be? Monolayer, yeah. You will not have a bilayer because maybe there's water outside, but inside there's no water. So the membrane will be lipid monolayer, okay. So these guys, oil bodies, have a membrane which is a lipid monolayer, not bilayer. Now if you look at plant tissue, there are three types of plant tissues. So three types of plant tissues. The first one is the dermal tissue, right? That's like the skin of the plant, right? So it's like the outer covering, you can say, like outer covering or skin of the plant. Then you also have what is called ground tissue, right? That's the kind of tissue that makes up the body of the plant. And again, I'm trying to just give you the overview. Okay, there are lots of details associated with those. Even in the ground tissue, you will have different, classifications and sub-classifications, right? And then the other kind of tissue that is important for us is the vascular tissue. That is the tissue that is going to be used to form these pipes, right, for transportation purposes that are called xylem and phloem. So vascular tissue, so xylem and phloem. So again, so you have two classes of vascular tissue, xylem and phloem, right? I will talk about these in a little bit more detail later on. But so xylem is basically the vascular tissue that will take nutrients and water from the soil, right, and it will give it to the other cells of the plant, right? So xylem, it carries water and nutrients from soil to shoot. And phloem carries material produced in shoots to the roots. And an example of that would be sucrose. That's the sugar that is produced in the leaves of the plant, right? And then the roots also need carbohydrate nutrition, so that flows through the phloem and is communicated to the roots, right? Now, one thing that you should keep in mind is that the xylem, right, it is basically a pipe that is made up of dead cells, right? The cells that were there have died, they have just left behind that structure, that pipe structure. So it's made up of dead cells, right? But in the case of the phloem, it is very much live, right? So it's made up of live cells. So live cells means there'll be activity, right? Some activity will be there, right? Different kinds of transport and all that. Dead cells means it's just a pipe, right? So the water pressure is going to push the water up the trunk of the tree. Okay, any questions? So that finishes our discussion of the first overview chapter, right? So chapter 1 is done. No questions? Okay. So chapter 2. Chapter 2 is about genome structure and gene expression. But this chapter is really very, very compact in that book, right? And fortunately, we have done these things in detail, right, in this course. So I can just, point out, highlight some important things. But again, I mean, this chapter, I don't think any of that stuff is just specific to plants, right? Most of it applies to plants and animals. Since we have discussed molecular biology in quite a bit of detail, I'm just going to fill in some of the gaps, some of the items that I did not discuss earlier, right? So the first thing that I want to talk about is epigenetic modifications, right? What are they? See, if you have an organism, right, you look at its DNA. Now, the DNA codes for proteins, right? So in order for, the protein to be produced, that portion of the gene has to be transcribed and then translated. It has to be expressed, right? Now, but even when the DNA looks the same, all right, the sequence looks the same, all right, the activity of the gene may be different, because if in some of the locations, let's say an A, all right, has got a methyl group on it, again methyl, remember, all right, or an acetyl group, that could alter the activity of the genome, even though the DNA sequence is identical, right, to whatever you had before, right. So, and these are called epigenetic modifications. They are not genetic modifications, right. So, and two examples of epigenetic modifications are methylation. Methylation, that means addition of a methyl group, right? And another example is acetylation. That means the addition of an acetyl group, right? Now, methylation, usually you'll find methylation in the case of DNA and also of histones. Anybody remember what histones were? They were the proteins on which DNA is bound, okay? So you can alter the behavior of the DNA through these epigenetic modifications. So methylation of DNA and histones, and then acetylation is only for histones. So you can either silence the gene, right, or make it more active just by altering the methylation and acetylation status, right, for number one. Okay. Now the second thing I want to introduce is just some terminology which I have not discussed before. There is a region, right, in the genome, right, that is called the nuclear nucleolar organizing region, right? Nucleolar organizing region, or NOR. And again, it's no big deal, but it's basically the region that codes for ribosomes. I just didn't want you to get thrown off if somewhere you read about this, right? The nuclear organizing region, right? Nucleolar organizing region. Another thing to note, and this is true for plants, is that plants exhibit polyploidy. That means they have many copies of the genes, right? It's not just diploidy. So you can have plants that are 4n, 4 copies of each gene, 8n, and so on, right? And they result from genome duplication. So let's say the cell is going to divide, right? Entire DNA has been replicated, right? But then the cell fails to divide, right? Because the entire genome is now duplicated, right? So you have more copies of the genes, right? So they result from genome duplication. Now, in addition, right, in addition to the nuclear DNA, right, the mitochondria and the chloroplast, they also have DNA in both in plants and in animals, right? So, so you also have organelle DNA. So, in addition to nuclear DNA, organelles like mitochondria and chloroplasts have their own DNA. And these are usually inherited only from one parent, right? For humans and all, it's only from your mother that you get your mitochondria, right? So usually inherited from only one parent, right? Therefore, the DNA that is there in mitochondria and chloroplast, that is not going to obey the Mendelian characteristics, okay? Because it's not like, the chromosomes split apart, then you get one copy from your mother, one copy from your mother, and then, from each parent, and then you get the combination of those characteristics. This is only from one parent, okay? So the important thing to note is that the organellar genetics, so organellar genetics, that means the genetics associated with these organelles like mitochondria and chloroplasts don't obey Mendelian laws. Like Gregor Mendel in the early 1900s, he did experiments involving pea plants and he was breeding them and all that. But with these traits that would be on these organelles, for those that's not going to work. Right? All right, any questions? So now I also want to talk about two other things. All right, one of them is RNA interference. See, when you transcribe a gene, all right, you get an RNA. Then that one is translated into protein, right? So if you want to stop after transcription, you want to stop the translation. What you could do is you could produce an RNA that is complementary to the RNA that you have just produced, and then the two will bind together. No transcription will take place, right? So RNA interference. So that's post transcriptional gene silencing, right? This can be done in the lab. You can use RNAi technology basically to stop a protein from being produced even after transcription, right? And in the cell also, there are things that are called microRNAs or really small RNAs that do this kind of thing. Like suppose some gene has been transcribed into RNA, but for some other reasons, for some other reason, the protein should not be produced. There could be a microRNA that is complementary to the RNA that has been produced, just bind to a small portion, so then the ribosome cannot get on and translate that, right? So that happens in the cell all the time. So I wanted to make you aware of that. Then also the role of ubiquitin, right? Remember, what is ubiquitin used for? The protein is basically used for? Mark it as trash, okay? It has to go into the trash can or proteasome, right? So again, same thing in plants and animals. So people learn something, okay? The exams were good, so now you are telling me things that you should have picked up and you have, which I'm happy to see, right? So ubiquitin tagging for protein destruction or protein degradation. So same thing in plants and in animals. All right, then I want to talk about techniques for genomic analysis, right? And we have covered some of them. So this chapter, of course, doesn't give any details, but we have covered some of these things in a lot more detail. So it'll just be like a review for you. So techniques for genomic analysis. First one, northern blotting, right? Again, northern blot, don't worry about the northern, okay? You have western blots, southern blots, northern. I don't even know which one is what, okay? But roughly speaking, all right? What procedure do you think they're talking about here? No, not you. You knew some of that stuff from before. It's somebody else. Based on what? And using what method? Gel electrophoresis. And then blot it onto that thing, okay? So you already know that, okay? So that's one, but that is going to be used only for one gene at a time. It's not a high throughput method, right? So you have northern blotting. Then you can also use RT-PCR. Again, RT-PCR is reverse transcription PCR, right? Right. So mRNA, do reverse transcription, you get the complementary DNA, then amplify many times with PCR, right? So these methods are basically when you're studying one gene at a time, right? Then if you want to do lots of genes at the same time, what methods should you use? No, that's sequencing, right? Let's say you want to study the activity of a lot of genes at the same time. Okay, and what else? I mean, I mentioned that before, I'll mention it again. This is called RNA sequencing, right? See, microarray, so microarray, what you do is you try to use the complementary sequence, right? And then get it to bind, look at the intensity and so on. But these days you can do sequencing very fast, right? So you will get the RNA, you will sequence it, right? Now, if that particular gene is being expressed a lot, you will have more of that RNA, right? So you'll have more counts, right? So RNA sequencing is, and again, the idea is this is high throughput sequencing, right? Or it's called next generation sequencing, where you take the RNA sequence, right, which is really long, okay, break it up into pieces that are 100 units long, sequence them, put them back together, there's assembly involved, right? So this is another technique that is used. This book mentions them, but I'm just like trying to give you the big picture. There are more details associated with it, okay? But I did mention that before, but Maybe we didn't discuss it in detail, so you don't remember, right? But these are methods that you can use, right? So these are high throughput technologies. All right, then the third one, which I have not mentioned here, is making use of what are called reporter genes. And again, these things are used both for plant genomics as well as for animal genomics also, all right? And here, this involves using green fluorescent protein or some kind of fluorescent protein. And the idea is very simple, right? What you're going to do is, let's say you are interested in monitoring the activity of a particular gene, right? Let's say there is a gene here, right? And you want to, so this is the gene, right? There's a gene A. You want to figure out the activity of gene A. Is it being getting transcribed, right? So this is the promoter region for the gene A, okay? What you could do is you could stitch this gene, right, together, and you know how to do that. You can use DNA ligase to do it. You could stitch this gene together with some other gene coding for a fluorescent protein. So you will put this, so you will put that gene A is under the control of some promoter. You will put this guy, the gene coding for a fluorescent protein, under the control of the same promoter. Control of the same promoter. So what is going to happen is every time that this gene is getting transcribed, right, even that green fluorescent protein is going to be produced, all right? So you're going to get light and you can track the activity of the cell, all right? So here you can do this without having to kill the cell. You produce what is called this reporter construct, all right, where it's basically reporting on the activity of that gene and based on the light intensity, right? The cell can be alive. You just put in that reporter, you can track it. You can basically track the activity of the cell, right? When does this one go up? When does this one go down, and so on. So this is also mentioned in that chapter, right? Any questions? Yeah.
Really more of a statement like how really cool that it is, like the jellyfish growth protein is a really good phosphorescent protein they use for this thing, and how it's like it's a really simple, small gene. It's really cool that it's expressed by everything, uniquely, very simple.
Yeah. But this is a non-destructive method, because if you're going to do microarrays to get the RNA from the cell, you're going to have to crush up the cells and then extract the RNA and so on. Okay. But this method is non-destructive because you can actually put the I mean, initially, of course, you have to use some kind of a vector to put that extra gene in. But once you have that, then it's like something that has been labeled, right? And you can track the activity. All right, any questions? And again, I didn't give you full details. Of course, there are details, protocols associated with these methods, but we are not biologists. We don't. I mean, if we are working in this area, there will be biologists that will be helping us with all this experimental stuff, right? So they'll take care of that. But this is the big picture. Now you can carry out, right, using the modern genetic engineering methods, you can carry out genetic modification of crop plants, right? For example, right, you can use that to modify rice in a way where the vitamin A content of rice is enhanced, right? Because there may be a region of the world where people are suffering from blindness because they don't get enough vitamin A in their nutrition, right? So you might be able to play around with the genes, right, in rice, right, and grow a variety of rice where the vitamin A contents has been enhanced, right? That's actually an example that I'm quoting from the book, right? Example: producing rice with enhanced beta carotene. Then in addition, using these genetic engineering methods, you can artificially transfer genes so artificially transfer genes to increase resistance to herbicides or plant pests or provide enhanced nutrition. which is what I mentioned about the case of the rice, so, or provide enhanced nutrition. Okay, any questions on this chapter? So again, it's an overview. Most of the stuff you already know, details of transcription, translation, combinatorial logic, all right, RNA polymerase transcription factors, all right, those are things that we have already covered. You've taken exams on that, so you know that stuff, all right. I don't need to go over that. And again, it's not even covered in this chapter in that much of detail in this book. So then that finishes chapter 2, right? We move on to chapter 3. So chapter 3 is about water and plant cells. Right? Now this business of water use by plants is extremely important, right? And actually in our center we have many students that are on AgriLife support. That comes from water use money, okay? Because in the state of Texas, let's say this year and last year it's a little bit different. You've had an excess of rains, but if you have drought, okay, how can you develop plants that are going to use water more efficiently, right? That's a big question. So there's a lot of money that is put into that kind of stuff, right? So if you're looking at plant water use, it is a fact that 97% of the water that is taken up by the plant is lost through transpiration from the leaves. So 97% lost through transpiration. Only 2% remains in the plant body. And 1% is used for photosynthesis. Right? So that means 97% of the water is effectively wasted, right? When you're trying to grow plants, right? And why is that? The reason is because for photosynthesis, the plants have to absorb carbon dioxide. Carbon dioxide plus water, right, is going to give you carbohydrate plus oxygen. They have to absorb carbon dioxide. What is the concentration of carbon dioxide in the air?
70%?
Carbon dioxide? No. It's about 0.4% or something like that, yeah. It's very, very low, right? So naturally if carbon dioxide is going to, and how is the carbon dioxide going to enter the plants? Through the pores in the leaves, right? The pores that are called stomata. Now the concentration of carbon dioxide in the air being very low, right? So the concentration gradient is not there, all right? At the same time, the water inside the plant, the concentration is high. So the way it works out is the water flows downhill, right? From inside the leaves, it will come out, right? And that is going to propel carbon dioxide going into the plant, right? And that is extremely, extremely inefficient because for one molecule of carbon dioxide to go in, the leaf has to expel 400 molecules of water, right? So 400 molecules of water lost for every molecule of carbon dioxide taken in. So it really makes sense, for us to focus on the properties of water to see if we can make this any better, right? This ratio, right? Now regarding water, I discussed the structure of water, right? When we were covering the molecular biology portion, we talked about all the hydrogen bonds and things like that, right? And there are a number of properties that came out of that. One of them is that water is an excellent solvent, right? For anything that is polar because of the interactions between the charges and the hydrogen bonding, all right? It's an excellent solvent for putting things in solution, right? Not for hydrocarbons, but for other things, all right? It's an excellent solvent. The second property of water is that it has very high specific heat. High specific heat means that despite the sun shining, right, if the plant is well hydrated, temperature is not going to rise that quickly, right? The temperature rise will not be that much, right? So that's a good thing. And water also has a high latent heat of vaporization. So this means that if water evaporates from the leaves, right, it's going to extract a lot of heat from there, which means cooling is going to be produced. Again, the leaf temperature is not going to rise that much. So those are all good things, right? Now another property of water that we want to talk about is surface tension, right? So it is the tendency, right? Surface tension is basically the tendency of water, right, at an air-water interface to minimize the surface area. Okay, so this will try to minimize the water-air interface. And so it'll be measured in units of energy, the energy that you will need to change the surface area, right? So energy per unit area, right? Which means the unit will be like joules per meter square. But again, joules is newtons multiplied by meters, right? I mean, force times distance. So Newton meters per meter square. So the unit of this will be newtons per meter inverse. Okay, just a dimensional analysis, right? Now, if you are looking, let's say at a curved surface, right? So if you're looking at a curved surface and you're trying to, so there is a, let's say an air bubble, all right, in water. You're looking at a curved surface. So inside this you have air, right? Now the surface tension is going to act along the tangential direction, right? It's going to try to minimize the area, and that's why this shape is going to be spherical, right? So the surface, but so if it's trying to minimize the area and it's acting tangential, right? That means the resulting force will be perpendicular inside. Okay, so net force is inward, right? So net force is inward. And the pressure that results from this force is given by this formula 2 times T divided by R, where T is the surface tension. Again, the units are consistent because for surface tension we said it was newtons per meter inverse, right? And then you have R over there, so it's newtons per meter square. So this is a unit of pressure, right? Again, these are things that you have seen in physics. I'm just going to try to relate them to plant biology, right? Now there are three other things that I want to discuss here: adhesion, cohesion, and contact angle. So cohesion means, if you're talking about cohesion of water, means the affinity for water molecules for each other. Okay. Adhesion means, let's say, the affinity of water for some other molecule, right? And the contact angle is what I'm going to define now. Now suppose you have a hydrophilic surface here, right? That means it loves water. If you put a molecule of water on this, okay, what is it going to try to do? It is going to try to make the contact area as large as possible, right? I mean, because it loves water. The water, this surface loves water. So typically, if you put in a molecule of water, it'll spread on the surface like this, all right? So this angle that you have here, right? The angle that is formed, this angle is called the contact angle. And if it's a hydrophilic surface, then this contact angle is going to be less than 90 degrees. Agree? So contact angle theta is actually less than 90 degrees. On the other hand, if it's a hydrophobic surface, right? That means a surface that hates water. So you have a surface that hates water. Then the water molecule will try to stay away from the surface as much as possible, right? So the molecule will look like this, right? I mean, and so the contact angle in this case is going to be this angle here, okay, which is an obtuse angle more than 90 degrees, right? So theta here is greater than 90 degrees. So now I'm going to use these ideas, all right, surface tension, then this hydrophilic, hydrophobic surface ideas, all right, to talk about capillary action, right? Now you may have done these in physics classes and all that, so this is like a review, and I'm not saying that mine is the greatest exposition. I've just picked up the bare bones to put the presentation together, right? So capillary action, so if you have a glass tube, right, and then you have, let's say, water here, right, then if you have a narrow glass tube, right, what is going to happen is that the water will move up that tube, right, and it'll form a surface like this at the top, right? Now, the contact angle here is this angle, right? You can see that, and again, the glass is something that loves water, right? So it's hydrophilic, hydrophilic surface. So the contact angle is close to 0. All right. Now, so because of this surface tension, all right, you have this curved surface here. Okay. Now, obviously, the surface tension is going to try to minimize that water-air interface, right? So there will be a force that is acting in the upward direction, right? That needs to be balanced by the weight of this column, right? Weight of the water column, right? Due to gravity, and that's the reason why this thing is going to move up the tube. Now, look at it this way: if the tube is narrower, right, that means the radius of that semicircle is going to be smaller. So that means the pressure is going to be higher, right? Which means you're going to need a higher column to cancel out the effect of the surface tension, right? So surface tension is upward. Balanced by gravity. And if you do the same thing using mercury or something like that, which doesn't like glass, right, you'll have a different diagram, right? But I'm not going to get into that right here because inside plants you don't have any mercury, right? So, right, I mean you'll have to put that in artificially. So the take-home message is that if you have a narrower tube, you'll get a higher equilibrium water height. All right, any questions so far? Next, I want to talk a little bit about the tensile strength of water. Right? So if you have a continuous body of water, right, and you start pulling on it, right, how much stress can it withstand before the body of water breaks, right? Right, that's the question, right? So let's look at a situation where, let's say we have a syringe here that is sealed, right? And I have water in there, and then there is a plunger for that syringe, right? Now, if there is an air bubble inside in this water, right? So if I push on the plunger, right, what will happen to the air bubble? It'll become, if I push to the left or on the plunger, right, what will happen to the air bubble? It'll become smaller, right? Because see, there are two forces at work here. One is the surface tension, right? That is going to try to make the bubble smaller, right, to reduce the surface area, right? And then whatever external pressure that you're applying, right? So if you're applying pressure towards the left, right, then you'll have this situation when you're pushing the plunger in, you have these external forces that will be acting inward, right? And also the surface tension will be acting inward because it's trying to reduce the size of the bubble, right? So this is pushing the plunger in. But if you start pulling, right, you'll see what happens, all right? And that's the interesting case. So if you're pulling means the external force is pulling away, right? But the surface tension is still inside, right? So in the beginning, let's say the pressure is balanced by the surface tension. You start pulling. So what is going to happen? What is going to happen, all right? The bubble is going to No, you are pulling it out, okay? So the force is acting outward. The surface tension is trying to minimize the air surface boundary, right? If you're pulling it out, you're pulling out the syringe, right? That means the force is acting on that boundary but in the opposite direction. So as you keep increasing that, what is going to happen? The bubble will become bigger, right? When the bubble becomes bigger, right, the radius will go up, right? What will happen to the surface tension? Huh? Decrease, right? If it decreases, then what is going to happen? It's going to grow even bigger, right? It'll keep on growing until the continuity of the water will be broken. Okay, you see that, right? So as bubble grows, so for the situation in the right, as bubble grows, continuity of liquid is broken, right? And so this is an example of what is referred to, like if you have surgery and some air gets in, right, you can get what is called an embolism, right? Right, there's a discontinuity in your blood, right? So it can be fatal. In a car, you can have what is called a vapor lock, right? In plants, you can have cavitation in the xylem. If there's cavitation, then water is not going to move anymore, right? So this is embolism. This is vapor lock in fuel line. And in the case of plants, it can cause cavitation in the xylem. Okay, any questions? So next we will look at different ways by which uh, you know molecules can move about, right? The first one is diffusion, right? Diffusion will take place in response to the concentration gradient, and the formula for diffusion is Js equals minus ds dcs dx, right? Where S is the solute that you're looking at, right? Js is the rate of transport, ds is the constant of proportionality, right? And this is the concentration gradient. And this is called what? No, ds is the constant, right, of proportionality. It changes with the solute, yeah. So yeah, concentration of the solute, yeah. This is the concentration gradient, right? So this is the concentration gradient, okay, because if there is a higher concentration of solute somewhere versus a lower concentration somewhere else, right, there is a concentration gradient. So that is captured in this, delta Cs divided by delta X, yeah. Oh yeah. The ds will depend on the solute. Yes, I don't know whether it depends on the density or not, but it will be different for each solute. Yeah, that is correct. Yeah. So this is called Fick's first law. And again, this is basically telling you that you know things will flow from a place of higher concentration to a place of lower concentration, right? And then some kind of equilibrium is going to be established. So diffusion is movement in response to the concentration gradient. And this is effective over small distances. So another kind of movement, right, is what is called osmosis. That is movement of water across a semi or a selectively permeable membrane. So suppose you have a situation like this, right? You have two solutions. On one side it is a concentrated solution, right? And then you have a dilute solution on the other side. And you have a semi-permeable membrane which does not allow the solute to flow through it. Okay? So what is going to happen? The solute cannot flow through this membrane, but water can, right? So if you have a concentrated solution where the solute concentration is higher on the left, then the water will flow from the right to the left. Right? In an effort to try and dilute the solution on the left hand side. Okay, the solute cannot flow, right? So this is a semi-permeable membrane, right? So this is a membrane. This is a semi-permeable membrane. This blocks solute movement. So water is going to flow from right to left, and that is called osmosis, right? You can have reverse osmosis, like if you increase the pressure on the, if you squeeze the solution on the left really hard, right? You can get water to flow from the right to the, from the left to the right, right? That is called reverse osmosis. And there are techniques for filtering water that use reverse osmosis, right? So reverse osmosis filtering is used to purify water, right? Not in the plants, but I mean, yeah, like Culligan, that company that supplies water, they do reverse osmosis filtering, right? That's by increasing pressure, okay? That brings me, right, to the point where I want to see the effect of water pressure, solute concentration, everything together, right? So we will need to look at what is called the chemical potential of water. So chemical potential of water, which is the free energy status of water. Now remember, according to the second law of thermodynamics, the entropy has to increase, right? Reactions will only proceed in a direction where the free energy goes down. Okay, remember that, right? So again, I mean, if you're talking about water flow, that is also going to be dictated by free energy considerations, right? So we are going to look at the free energy of water to determine in which direction it is going to flow. So we are going to look at free energy of water. But plant biologists usually look at free energy per unit volume, right? So free energy per unit volume of water, and they refer to this as water potential. And what is the unit of that going to be? See, free energy, what is the unit of energy? Joules, okay? So it's basically force times distance, right? And then if you're looking at volume, that is basically as the cube of the distance, right? So cube of the distance. So what is the unit of this going to be? What? Pressure, yeah. This is going to be pressure units. So the water potential is going to be specified in pressure units like Pascal, megapascal, and so on. Okay? Now water flows from higher chemical potential to lower chemical potential. And if you're looking at water potential, it has got three components, right? So water will flow down the water potential gradient, and the water potential has three components. Psi is made up of psi sub s plus psi sub p plus psi sub g, right? Psi sub s is what is called the solute potential or osmotic potential. Psi sub p is what is called the pressure potential. Remember I told you you could make water go from one side to the other by applying pressure, so that plays a role too. right? And then psi sub g is called the gravitational potential. Okay, let's try to understand what each one of them is, right? Now psi sub s is the solute potential. See, if you add some solute to water, you're diluting the water, right? So you're making the potential negative, right? And in fact, psi sub s is given by this formula. Psi sub s is minus r T times the concentration of the solute. So R is the universal gas constant, Cs is the concentration of solute. So if you stick some solute, if you dissolve it in water, you're lowering the water potential, right? Because then if you have some free water somewhere, it's going to try to get in there. Okay, you lowered the water potential. Psi P is the pressure potential, right? So if you apply positive pressure, right, you're going to raise the potential, right? If you increase the pressure, you can force the water out, right? So pressure potential, if you have positive pressure, that's basically like compression, right? And this is, in plants, it's also called the turgor pressure. That can be built up as the cell is trying to grow against the cell wall, right, with all that water in it. That's turgor pressure. On the other hand, if you have negative pressure, that is called tension, right? And these pressures are measured with respect to the atmospheric pressure. So if it's one atmospheric pressure, then it will be zero, right? The potential is going to be zero, right? And then the gravitational potential is something that all of you know from physics, okay? If you have a column of water that is of height h, the pressure is going to be density times g times h, rho gh, right? You already know that. Now it turns out that that gravitational potential portion you can ignore if you're looking at the cell level, okay? If you're studying cells, right? I mean, by how much is it going to change? They're essentially at the same height, okay? It's micrometers or whatever. So you ignore that. So usually the relationship that is used is psi equals psi s plus psi p. So the solute potential plus the pressure potential, that's the total water potential, right? Yeah, just two more things I need to mention and I'm done, okay? So we note that because of the rigid cell walls, all right, small changes in volume, in cell volume, lead to large changes in pressure, large changes in turgor pressure. Right, and then also there are some membranes that are called aquaporins, right, that are water selective pores formed by integral transmembrane proteins. And finally, we note that solute accumulation can lower water potential. And this happens in plants. Suppose the water is not flowing into the plant from the soil, okay? What does the plant need to do? It needs to lower the water potential. So it will start producing solutes, right? Water potential goes down, then the water is going to come from the soil. Okay. So that finishes chapter 3. I had hoped to even cover chapter 4, but I'll do it next time. Again, I mean, this is the first time I'm doing this, so I really have no idea about how much I can cover in one lecture, right? But I mean, we'll make sure that it's something reasonable, right? So that's all for today.